Alcyomics is a contract research organisation specialising in predicting adverse immune reactions to compounds, including pharmaceuticals and cellular therapies. From my work in the bone marrow transplant field on predicting a serious complication of transplantation, graft versus host disease, I had the idea to develop an assay to predict adverse immune reactions to new drugs and compounds, including cellular therapies. With this idea, Alcyomics was born and started trading in 2009. We patented the new assay in the US and Europe and have had contracts with some of the top 10 pharmaceutical companies, helping them to ensure their new drugs are safe. We trade worldwide, including Japan, Europe, and the USA. Our work primarily feeds into the manufacturing and preparation work package at the Northern Alliance Advanced Therapies Treatment Centre, which has been a conduit for introducing our safety assays to the cellular therapy community. An adverse immune response, or immunogenicity, in the cellular therapy field means an immune response caused by the cellular therapy product which causes harm to the patient. This could be a skin rash at the site of injection, or more severe systemic response, known as graft versus host disease, in some cases where the incoming cellular product attacks the tissue of the patient instead of curing the disease. This can occur when the product is from another donor other than the patient themselves, an allogeneic response, and when the product contains cells not specific for the target. In addition, concerns were raised with regard to rejection of the cellular therapy product. When a product induces an adverse immune reaction, the cells can release cytokines or other molecules that can cause tissue damage. Assays for these cytokines can be tested in the lab and are known as T-cell proliferation or T-cell activation assays, which may indicate if the product may cause an adverse immune event and off-target responses. At Alcyomics, we have developed not just cytokine release assays, but also assays which can show the direct tissue damage caused by the cellular product. Here we obtain blood and a small 4mm punch biopsy from healthy volunteers with informed consent via our NHS research nurse who collects the samples. These are processed in the laboratory and assessed or graded in order of severity for tissue damage, indicating both the safety and efficacy or degree of off-target non-specific reactions which may occur following therapy. These assays are part of Alcyomics' Skimmune or skin explant platform. Within the NAATTC partnership, Alcyomics has worked together with the Newcastle University and Scottish National Blood Transfusion Service and tested novel products for safety and efficacy. In this regard, we have tested new anti-COVID-19 drugs consisting of T cells specifically directed at killing COVID-positive cells for use in clinical trials against COVID-19. Our assays have shown that these cells are specific and demonstrate very little tissue damage in our assays. Similarly, we have tested specialized cells directed at down-regulating the immune response to rheumatoid arthritis and have equally shown that the cells could cause minimal or no damage to tissues. These results are encouraging and demonstrate that Alcyomics assays are useful tools for assessing the safety of cellular products prior to clinical trial. MHRA approval is necessary before any product can be administered into patients. This normally starts with a Phase 1 safety trial by recruitment of healthy volunteers. Prior to Phase 1, safety checks of the product are needed to be carried out, which include stability, risk of cancer induction, adverse events, sterility checks, production reproducibility, and many others, which need documenting and submitting to the MHRA as an investigative medicinal product dossier. Alcyomics has published widely on the use of their safety assays, and the use of them within the cellular therapy field is important recognition of their potential importance. We hope that the assays will aid in allowing more cellular therapies to get to the patient quicker in a more timely manner in the future.